All right, so thank you everyone for joining us here today. This is the first Biochemistry Colloquium of 2024. So yeah, we're getting off to a great start here with Xiaobing Shi, Dr. Xiaobing Shi. I am delighted to introduce you today. I've known him, Xiaobing, since, I don't know, I think 2015 when we used to see each other at meetings, and more recently at the NCI study section where we're both members. So Xiaobing is currently a professor in the Department of Epigenetics at the Van Andel Research Institute in Grand Rapids, Michigan. Xiaobing is from China. He received his BS in biology from Wuhan University, and he traveled to the Chinese Academy of Sciences for his graduate training, where he focused on the mechanism of ATP synthases in chloroplasts, and he graduated with honors from both institutes. He subsequently pursued postdoc training at Stanford University with one of the fathers of modern biochemistry, Arthur Kornberg, and he studied inorganic polyphosphates and their function in affecting sporulation and biofilm and motility of B. cereus. So following a successful project in the Kornberg lab, and he closed down the Kornberg lab. I think he was his last postdoc in Arthur's lab. He joined a junior PI by the name of Or Ghazani, who many of you know from chromatin field. Xiaobing was Or's first postdoc. And Or had recently established his lab and was interested in researching domains of lots of chromatin-associated proteins. And Xiaobing had one of these like dream postdocs where he uncovered the fact that PhD fingers are recognizing methylated lysines of monomethyl, dimethyl, and trimethyl, and they can distinguish those different methylated states. Xiaobing, before he joined Van Andel, he went to the University of Texas MD Anderson Cancer Center, where he held the position of associate professor and served as a co-director of the Center for Cancer Epigenetics until his move to Van Andel. His lab has had many outstanding contributions to the field of chromatin and cancer biology over the years. They identified a 3.3 specific reader, ZMIND11, which has roles in splicing and gene expression. They discovered that the Yates domain is a reader of histone acetylation, not only acetylation, but also crotonylation. And more recently, they've connected dysregulation of reading and writing histone acetylation to different cancers. For example, ENL, misregulation of that in AML leukemias. So his lab is delved into this intriguing regulatory mechanisms that affect involving lysine acetyltransferases, and I think we're going to probably hear more about that today. Xiaobing has been honored with several distinguished awards, including the Career Development Award from the Leukemia and Lymphoma Society, a Research Scholar Award from the American Cancer Society, a Kimmel Scholar Award, and recognition as the inaugural R. Lee Clark Fellow from MD Anderson. So please join me in welcoming Xiaobing today.
Thank you, Peter. Can you hear me well? For the nice introduction. I wasn't aware this is a biochemical series of seminar. I regard myself as a biochemist, but in front of the real biochemist, I'm a little bit nervous to present biochemical data. Yeah, probably you want to ask me more about epigenetic question rather than biochemistry. So I try to do my best. I feel a little bit ashamed saying that I did my postdoc with Arthur Kornberg, who is regarded as one of the best biochemists in the world, right? So I come from Van Andel Institute. If you don't know, Van Andel is just crossed Lake Michigan, the next side, the other side of Lake Michigan. So we have similar weather as here. So I'm not afraid of weather here. So we know there are a few mechanisms that regulate gene, epigenetic mechanisms regulate gene expression. There are DNA methylation, right? What we know is promoter DNA methylation is associated with gene repression. There are post-translational modifications on histone proteins. There are also histone variants coming with different taste of histone sequence and histone chromatin remodelers. Among these four mechanisms, my lab is mainly focused on histone modifications, actually mainly focused on histone methylation and acetylations. So when we talk about a particular modification on histone, for example, normally we talk about three family of regulators. They are writers, readers, and erasers that we call them, right? Writers are the enzyme that put on this epigenetic marks. In case of methylation, it's KMT, lysine methyltransferase. And in terms of acetylation, the CAT, lysine acetyltransferase, or HAT, histone acetyltransferase. The erasers are the KDMs and HDACs, and the reader proteins are actually the domains that recognize specific modification in a sequence, most likely in a sequence-dependent manner. So my lab started with interest in the reader protein, as you heard from introduction from Peter. Because it's the discover or research interest switch, we start to look at writers of these modifications, as we're gonna see along the talk. So this slide summarizes most known readers of histone methylation and acetylation. As you can see, for example, here's the chromodomains. That's the first discovered reader of histone methylation. It's that HP1 protein. Right, chromodomains, they mainly recognize the repressive mark, trimethyl species, right? K9 or Kden7 methylation. Also, PWP domain recognize the different methylation on different active mark, we say, on different residues. And there are different readers recognize slightly differently as lower species of methylation, such as a Tudor or MPT domains, where there are also other domains, right? If you, Peter introduced to you that I stepped into this field by discovering that the PhD finger as a reader of histone A3K4 trimethylation, that's the into PhD finger that I published more than 20 years ago, less than 20 years ago. And later on now, we know there are at least two families of PhD fingers. One is the into type, which recognize A3K4 trimethyl. The other is BHC80 type, which recognize A3K4 amine0, meaning that it binds to this unmodified histone A3 tail, but the binding is disrupted by methylation on these residues. So over the years, my lab has been collaborating with different labs and characterizing different PhD fingers. And we also try to identify or discover new readers of histone modifications. One example is the PWP motif of the protein called ZMN11, which recognizes A3K36 methylation in a histone variant specific manner, 3.3. And also we discovered a second family of histone acetylation reader. So if you look at this slide, right, most of the reader domains, they recognize methylation, but with one exception is a Brahma domain, probably many of you are aware of, which bind to acetylated lysines. And Yates domain, we find that they also recognize histone acetylations. And here, there are four Yates domains in human proteome, F, and I, E, and L, Yates2 and Gas41. The first two proteins, they are components of two complexes. One is superelongation complex, the other is .1L complex, which deposit methylation on A3K79. And both complexes, their function is to promote transcription at elongation stage. Another Yates domain protein, Yates2, is part of the GCN5 containing ATAC complex depositing acetylation on histone A3, and Gas41 is part of TIP60 complex acetylation histone H4. It's also a component of SORCAP complex, which deposit histone variant H2AZ into the chromatin. So by using different assays, we demonstrated that all the Yates domains of human Yates protein and of yeast as well they bind to histone acetylation mainly on histone A3, with the preference on acetylation on A3K9, K18, and K27. If you look at this sequence, right, the common feature of these three is the archaic motif. You have the arginine prior to the acetylated lysine here. So that explains the specificity, with the only exception is the GAS41, which can also recognize GK motif. So through collaborating with Haitao Li's group at Tsinghua University, they saw the crystal structure of all the human yeast domain. As you can see here, right, all the domains, they have similar structure, which is a kind of a sandwich structure of 8 beta sheets. And the recognition of acetylation is achieved by this deep hydrophobic pocket formed by this 4 or 5 aromatic residues. And there's also a hydrogen bond between the serine and the acetylated lysine here. So, by looking at comparing the study from my labs and many others, we probably can distinguish yeast domain from Brahma domains, right? But although both are readers of histone acetylation, there are different features. For example, the yeast domain, they have deeper pocket. It's actually not only a pocket, it's a channel that the peptide can insert into. Because they're deep in the size, so it's not only recognized acetylation, it's also recognized different acetylation that with a longer side chain, such as chromatodylation, propionylation as well. And as a comparison, Brahma domains, they are much shallow pockets and much wider. They can only accommodate acetylation, and because they're wide enough, some of them, they prefer diacetylation. You can accommodate 2 acetylysines in one pocket. And as I just mentioned, the yeast domain, because they prefer the archaic motif, and because the sequence more frequent in histone 3, so yeast domain, we can regard them as readers of A3 acetylation. Now, Brahma domain, as in contrast, they prefer the small neighboring amino acid, or they have no strong preference. They mainly care about acetylated lysine. And that's why they bind to all acetylysine histones, and with the preference of histone H4 acetylation. Most of them. Of course, there are some certain exceptions, right? So as I just mentioned, because of the research proceed, for example, 2 of the yeast domains associated with HAT complex, right? With yeast2 is part of this complex, and GAS41 part of this complex. We start to examine the different acetyltransferase writers, right? And the more we look into, the more interesting they are to us. For example, the GCN5, they're specific for acetylation A3K9, and also acetylate A3K4, and mass morph acetylate A3K23, and P300CBP acetylate K27, and K18 on histone A3. And these 2 enzyme complexes, they deposit acetylation on histone H4. So for today's talk, I'm going to focus on these 2 enzymes, right? The homologs to each other, P300CBP. So what are P300CBP? You can ask ChatGPT to give you this list of quick answers. It's largely right. But there are enzymes, histone acetyltransferases, that deposit acetylation on histone A3, K27, and K18. They actually are quite promiscuous. They can acetylate the other histones. But again, K27 and K18 are one of the best markers we can use as indicator of their enzymatic activity. Because they acetylate histone, they open up the chromatin, so they are naturally transcription co-activators. They promote gene activation. They are essential genes. They're essential for development. So homozygous knockout of CBP or homozygous knockout of P300, or heterozygous knockout of one allele of each are embryonically lethal. They're also frequently mutated in human cancers. That these two proteins, they are probably one of the most frequently mutated genes in human cancers. So this is the sequence of histone A3 protein, P300CPP proteins. As you can see, they are large proteins comprised by different well-defined protein domains and linked by these non-structured regions. The distal domains, such as nuclear receptor indexing domain, TAS1, KIX, TAS2, and IBID domains, they've been identified as motifs that bind to different transcription factors, as I listed here, that many of transcription factors, probably hundreds of them, that P300CPP can bind. But probably the most important domain, as I can say, of this protein is the catalytic domain, the head domain, which catalyzes acetylation on lysine residue. You can see here that there are a few other domains, just next to this head domains. For example, here's a Brahma domain, as I just mentioned, recognize histone acetylation, and this recognition actually promote the head activity. And here is the ring PhD finger fused domain, and this domain has a structured role that interfere with the head activity, actually has an active role regulating head activity. And here is AIL, stands for auto inhibitory loop. From the name, you can tell it has a inhibitor regulation role of head activity. That's the AIL. So besides this, there's also ZZ domain and TAS2 domain. And for today's talk, I'm gonna focus on discoveries of the function of these two domains. So this is a structure of P300 head domain together with Brahmo and ring finger. It turns out the three together here are, they form quite stable structures. This has been solved by several labs, and this is the first structure solved 10 years ago with blue color represent the head domain, and here the green one is the ring PhD finger, and here's a Brahma domain. And this Brahma domain here is the pocket for acetylation recognition, and here is the catalytic region, a pocket for substrate acetylation, and there's an auto inhibitor loop, which is unstructured. That's why you don't see that in crystal structure. We guess that probably somewhere around here, that fall back into the catalytic site, block the catalytic site from the substrate. That's the apo structure. If you put this on nucleosome, the structure itself remains stable, but the orientation on nucleosome could be quite flexible. For example, the Brahma domain here, they bind to H4 tail there. In this case, with the head domain point towards the A3 tail, and here probably towards the H2B tail. So this explains why this enzyme can catalyze acetylation on multiple histone tails. And if I remember correctly, this structure, they started with the whole region here, but because these two regions are so flexible that they cannot see in the crystal structure. So the ZZ domain actually we identified is functions as a reader of histone A3 tail. The reading activity is unique. It's different from other readers that we know of. And here I show, if you compare P300ZZ to RBP2 PhD finger, this is also PhD finger, a reader that recognizes unmodified histone A3 tail, but this binding is abolished by methylation on K4, or arginine on R2. However, this ZZ domain of P300, it doesn't care about this modification, whether it's methylation on K4, methylation on R2, or acetylation on K4. They have similar binding affinity to histone A3 tail. This can be explained by the structure, which is solved by Tanya's group, Yijan, who was a postdoc in Tanya lab at that time. So this is the ZZ domain of P300. and the blue stick represent histone 3 tail. And if you compare the structure to a known rater of unmodified histone 3 tail, we can sort of tell why they are different, how they are different, right? In this case, the R2 and K4 is inserted into kind of an open space. However, in this case, both residues insert into kind of a small cavity, which limited the size of the side chain. So that if you imagine you have a methylation there, you're gonna interfere with the amino acid around this pocket. So that explains why this domain is sensitive to modification on these residues, but this domain does not. So based on the structure, we can mutate the, I forgot to mention that these two are not that essential, right? The most essential residue is alanine 1 here that form a few hydrogen bond with a few amino acid around there. So based on structure, we can mutate amino acid that interacting with the alanine 1. In this case, these two residues. We mutate to alanine, you can abolish the binding. And point mutation of these residues, you reduce the activity of P300 hat towards K27 or K18. This is the in vitro hat assay. You can also see reduced histone acetylation in cells. So basically, we can conclude that in addition to the other positive or negative regulation of this domain, so Z domain here positively regulate hat activity by recognizing the unmodified histone A3 tail. So I just want to show you this slide that by that time we didn't have the structure of the whole thing, but we do have the structure of this 3 domain together and the Z domain, and the E actually predict based on this different characters that the Z domain likely stand around here and recognize histone 3 tail represent the rest of histone to the catalytic domains. In this case, I think here's K18. And later on, this being proved probably correct, right? Because this is still a model, but predicted by AI, right? By AlphaFold, they predict the Z domain likely reside in around similar location here. But still, we need structure biologists to tell us by real structure that whether this is a real or not. Right? So, as I mentioned, right, P300 or CBP is one of the most frequent mutated gene in cancer. And here I show P300, and CBP actually is similar to this, right? P300 gene is mutated. You have more than 2,000 mutations across more than 100 type of cancer types. And we accept a few mutations, right, enriched in the hat domain, and we know these mutations, most of them are loss of function mutation, meaning that the mutation, right, kills the catalytic activity. But there are so many others, right? Thousands of other mutations. I guess the question is, which mutations are driver mutation of cancer, or many of them are, just passenger mutations, right? So how to address this question? You probably can catalyze each of them, but that will take probably forever. We can utilize the CRISPR technology, right, to mimic the mutation in human cancers. Here we use the CRISPR Titan array by collaborating with Han Xu's lab at MD Anderson. So basically, you design guide RNA as many as possible targeting your gene of interest, right? You cover the entire coding sequence by frequency about every 8 nucleotide, you can find a guide RNA. So by that frequent coverage, right, you're screening the essential cell line, right? The CRISPR Cas9 have, the cut has 2 consequences, right? It's gonna be repaired by DNA damage repair pathway. You can have either in-frame mutation or frame shift, right, mutations. If it's frame shift, then you're likely gonna kill the protein. Then you got an inactive protein, then you kill the cell. But if it's in-frame mutation, in a non-essential region, likely the protein is gonna be still active. And if it's on the essential region, you're going to kill the protein as well. So by this principle, if you have enough guide RNA, you can precisely predict which domain is essential for the protein function. And that's the principle of CRISPR tiling array. So by using this technique, we screen for different enzymes. In this case, I show you P3 and CBP. So I forgot to tell you that in most cancer cell lines, P300 and CBP, they are kind of redundant to each other, meaning that if you knock out P300 cell are totally fine because they have the wild type CBP there. However, in a cancer cell line such as RKO cell we use here, which is P300 null, then CBP become essential. So if you have a guide RNA targeting the essential domain of this function, right, you leads to loss of function mutation, then you kill the cell. The negative score of the Z-score here indicating that the cell died. By this way, we can identify a few essential domains of CBP, right? You can see here's a Brahma domain, and most of this hat domain, and a little bit of region over there too. So at the same time, we also used wild type cells, P3 and CBP wild type cells. We use that kind of as a negative control. We didn't expect that knockout of any of this is gonna cause cell death. But however, we noticed that for both P300 or CBP, if I get RNA targeting this region, the red box region, you actually kill the cell. And this region is the TAS2 domain of these two proteins. And is that real? So the first thing you want to do by for any kind of screen is to validate the screen, right, by individual assays. So we design a guide RNA targeting TAS2, and we also design a guide RNA targeting different regions, hat domain, and the domain just upstream of TAS2 ZZ domain, and domain downstream of TAS2 IBID domain. And by this individual guide RNA, you can see, right, only if you target in the TAS2 domain, it causes more severe cell deaths, which validate the screen result. So I forgot to mention, maybe I should go back to previous slides here that, so think about this. If I have guide RNA targeting the hat domain, which is a loss of function, does not kill the cell, but guide RNA targeting this domain, then they should not, this kill the cell, they should not be loss of function, right? So probably it's a gain of function mutation there, right? So that's the hypothesis. So then is that gain of function? So here we look at a protein first, or P300 or CBP. Here's a control, and this TAS2 knockout, it can lead to mostly truncation and a few probably indel deletion mutation there. And hat domain, as we know from previous study, that because the folding of the hat domain is so essential that if you alter any amino acid sequence there, you destabilize the protein got degraded. And other guide RNA also lead to expected truncations. Are they gain of functions? I think the short answer is yes, they are. If you look at the target residue acetylation, right? In this case, look at K18 acetylation or K27 or Histone 3. Only in TAS2 truncated cell, if compared to control or other guide RNA treated cells, you see increase of acetylation, but not other residues. You can see the same thing by CBP TAS2 truncation. And those are guide RNA added cells, but this phenotype we can observe actually in natural cancer cell lines that contain this TAS2 truncation. In this case, we use this OBCR5 and 8, which contain TAS2 truncation. You can see massive increase of oxidation compared to P3 and CBP wild type cells, right, on K18 and K27. And actually, CCLE also did mass spec analysis of Histone acetylation on more than probably 600 cell lines with probably around 10 to 20 cancer cell lines containing TAS2 truncation. And if you look at this peptides contain K18 acetyl or K27 acetyl, you have massive increase of acetylation. if cells contain P3 and CBP test2 truncations compared to P3 and CBP wild-type cells or other truncations, indicating, again, demonstrating that if you truncate test2, then you increase global acetylation. So where are they? So we did ChIP-seq analysis of, in this case, cadherin-7 acetylation. We divide the acetylation into 3 parts, right? Gene body, promoter, and intergenic regions. You can see acetylation increase in every regions we look at, more prominent in gene body and the promoters. You can also look at the peak densities. In this case, we look at A3 cadherin-7 acetylation in the cells. The y-axis represent the test2 truncated cells. Here, the x-axis represent the control cell lines. You can see that overall the dots shift towards the upper left, indicating that there are increased density of Ka27 acetylation in the knockout cells, right? This is one guide RNA, this is another guide RNA. And the control cell head knockout, you don't see changes, right? See slightly more there, which is natural because you knock out the enzyme, right? If you look at the heat map, you can see the increase is not only in the center of the peak, but also in the nearby regions. In this case, we look at quite a wide window, actually. It's 20K up and downstream of the peak. You can see increase everywhere in this test2 SG1 and test2 SG2, right? Here's the average profile. Again, you can see increased not only in the peaks, but also in the flanking regions. So acetylation, we know their general function is to open up the chromatin and to promote gene activation. Is that the case in the test2 truncated cells? In this case, we look at the fold change of acetylation and their correlation with fold change of gene expression. This is knockout test2 truncated cell over control. We look at acetylation of cadherin-7 at gene promoter, and you can see largely correlated. And here, the red dots indicate that the genes with increased acetylation at their promoter and increased gene expression, right? You can see in both test2 knockout cells. And this correlation, you can also see if you plot increased gene acetylation in gene body, right? And they nicely correlated with gene expression. So here I show one example of P21 gene. You can see here the control cells, 3 biological repeats. You have a very mild acetylation there, but with test2 truncation, you have huge increase of acetylation in the promoter and across the gene body, right? One guide RNA, the second guide RNA, and here's a head SG control. And here is gene expression, right? With increased acetylation is to increase gene activation. And by looking at global gene expression, we can see 3,000 gene upregulated and the 2,000 gene downregulated. And here's the terms enriched in the upregulated genes. You can see apoptotic genes and also ECM and cell adaptation genes over there. So I guess as a biochemist, I'm interested in how test2 interact with or inhibit head activity because they are close in physical distance. I guess probably there is a physical interaction between these two domains, right? With here's the head, here's the test2, you're likely gonna be physical interaction and the kick off the head activity. So is that true? So we can test this by a few biochemical assays in vitro. So we cloned we call it core of P300 protein, right? Which include from bromodomain downstream up to this test2 region, right? This core. And then within this core, we delete the test2, right? Delta test2. We purify this protein from cells and we catalyze in vitro head assay. So the core with test2 there is a kind of inert enzyme. You can acetylate substrate nucleosome, but the activity is quite weak, right? Within 30 minutes of reaction. However, if you remove the test2, once a sudden become very active enzyme, if you compare this two. We can also see this drastic difference by using CBP core with the CBP core delete of the TAS2 or superactive enzyme with TAS2 truncation, indicating that TAS2 is has a negative role regulating the head activity. We can also do a few, this enzymatic assay. It's a very simple assay that we titrate different nucleosome into the P300 core or core delta TAS2. If you look at reaction speed, right? This is the core, and this is the one without TAS2. It's a much better enzyme with lower Km and higher Kcat. And you can, this is on K18, and this is on K27, right? When you remove TAS2, it becomes a much better enzyme with higher affinity to the substrate. So the TAS2 structure is known, it's been solved probably more than 20 years. It's a domain comprised about 100 amino acids and forming this 4 alpha helix, 1, 2, 3, 4. And if you look at cancer mutations, here show the P300 CBP. Here I only look at the truncation of these 2 proteins within this region. There's no hotspot, right? The truncation could occur on every alpha helix here. So we took a few. I think here I just show you one. That one, all these 3, they are cancer mutations. This one represent the full truncation of the TAS2, and this is the downstream of TAS2. You still retain the entire TAS2, and this is the truncation within the 3rd alpha helix, and this is actually a mutation occurring in the OVCR5 cancer cell lines. And then we purify this truncated cells, and we carried out in vitro assay. You can see here, again, the full length is a weak enzyme, and if you truncate out even in either in the entire TAS2 or partial of the TAS2, it becomes superactive enzyme, right? There's a short exposure, long exposure of K18 or K27. You can see huge increase of the activity compared to the full length proteins, right? Indicating again, if you don't have the TAS2, the enzyme become a superactive enzyme. And here we look at autoacylation. You can also see huge increase of the autoacylation of the enzymes. So if you remember from the very first few slides, I mentioned to you that the TAS2 domain has been regarded as a domain that interacting with transcription factors. Actually, the structure have been solved of TAS2 with several transactivation domains. In this case, I show you the structure of TAS2 with the P300 P53 TAT, and with P53 here in the yellow ribbons here. So then the question is that 2 functions of TAS2, one is inhibition of HAT, the other is binding to transcription factors, the TAT. Then are these 2 functions linked to each other, affect each other? So we can test this by titrating different amount of P53 protein into the in vitro head assay. In this case, here's we used the P300 core incubated with nucleosome. We titrate in different P53. You can see with the increased amount of P53, you can see increased oscillation of substrate histone, in this case, K18 and K27. So when you add P53, that doesn't affect the TAS2, the catalytic activity of the core without TAS2 because itself is already superactive enzyme. You don't see further increase, or maybe because you don't have TAS2 there, P53 doesn't bind to this region. So this P50 dependent activation requires the TAT motif, require this region. If you delete the TAT, there are actually 2 TATs. I delete both TATs of P53, you no longer can see the activation of P3's catalytic activity. And this binding dependent activation of P300, you can see that not only with P53, but also a few other transcription factors that bind to TAS2 motif. In this case, we tested FOXF1, CBP, BMIP, P63, and then STAT1. All of them can more or less enhance the catalytic activity of a P300 core. And it has also been reported recently that the NUT transcription factor can also activate P300. So with this, we can come up with a simple model that in apo state, when the enzymes stay inactive, so P300 adopt a few mechanisms to remain closed or inactive. One is AIL to block the pocket and ring PhD finger. It also has a structural role to block this region. And also you have the TAS2 domain here, probably somehow interfere with substrate accessibility here. However, if you have transcription factor coming over, it binds to TAS2. So this binding not only recruit P300 to target regions where the TF binds, but also open up the enzyme so that the catalytic pocket is accessible to a substrate such as histone 3. And then with this open, this got acetylated, this further open up. And then the ZZ domain recognized histone 3 tail, further stabilized the substrate at this region. And also there's actually a Brahma domain bind to acetylation 4. It's also stabilized the substrate to the enzyme. And in cancer cell lines, if we have TAS2 truncation, then basically the enzyme become constitutive active. So you have global increase of acetylation. So now we have the scenario that in cancer cell lines that contain TAS2 truncation, you have increased acetylation, and the increased histone acetylation leads to likely contribute to cell death. So this is very similar to another scenario that we are familiar with, which is what? Which is actually the strategy we use to kill cancer cells, right? We use HDAC inhibitor to treat cells. We induce global acetylation, and that lead to cell death, right? And then the question is, because both increase histone acetylation to lead to cell death, whether there's a synergy between these two, or whether the cancer cell lines that contain TAS2 truncation, whether they are more sensitive to HDAC inhibitors, right? That's the question we wanted to ask. So we can test this by, again, using the cancer cell lines. We generate TAS2 truncation along with a few other truncation as control. And we treat cells with different amount of concentration of HDAC inhibitor, SAHA. You can see here, right, the red line represents TAS2 truncated cells. They are more sensitive to HDAC inhibition, right? If you look at the GI50, right, in these cell lines, the control cell is about 3 in this cell line as well. Then if you have TAS2 truncation, the GI50 is reduced to about 1 or less than 1 micromolar. We can see this not only with SAHA, but also with another HDAC inhibitor. Right? This is a FDA-approved HDAC inhibitor, panobinostat. We can see the same thing. We can reduce sensitivity from 12 nanomolar to 2 or 3 nanomolar, right? It's a huge increase of sensitivity. And the same thing that if you generate a CBP truncation, you can also see the same thing. So it doesn't matter whether it's CBP or P300, as long as you have truncated P300 at this region, you have overactivated enzyme, you have global acetylation, the cell becomes sensitive. So is that because of global increase of acetylation? So the answer is yes, right? Here you can see by looking at this K18 or kinetic acetylation, you treat these control cells with different increased dose of SAHA. You can see, right, gradual increase of acetylation. But in the TAS2 truncated cell, you have a huge amount of acetylation there. And this is further increased by SAHA treatment. Right here you can see the same with another HDAC inhibitor. So the synergy you can see not only by global acetylation, but also you can see that by G expression, right? In this case, you can see here the control cells, if you only treat with SAHA for control cells, you can see increase of acetylation, increase of gene expression, but the increase is not that drastic. However, if you have test to truncation in the cells, you have a drastic increase of gene expression compared to the control cells. Right? There's a one SG, there's the other SG, right? With more gene being upregulated, with fewer genes being downregulated. So this is probably the last data slides I have. So we can see this synergy not only in vitro, we can also see this in a xenograft mouse model. In this case, we generate xenograft using control cell lines or cell line containing test to truncation, and they treat mice with SAHA. You can see here that control cells, they are less sensitive to SAHA treatment, but the test to truncated cell, they are more sensitive, right? And this is sensitivity observed not only by artificial CRISPR-generated knockout cells, test to truncated cells. You can also see in cancer cell lines. Again, we compare this to cancer cell lines, which contain test to truncation over other P3 and CBP wild-type cells. Basically, you can see these cell lines that are more sensitive to HDAC. So with that, today I showed you two more regulatory events of P300 enzyme. One is the Z domain, which is histone A3 tail reader that has a positive role regulating the head activity. The other is the test domain. In addition to be a TF binding motif, it has a negative role regulating the head enzyme, and the cells contain test to truncation. They are more vulnerable to HDAC inhibitors, right? Providing further strategy for treating this type of cancers. So with that, I'd like to thank people who contribute the work. The P300 part was carried out by two postdocs, Long Xia and Hong Wen, these two here. And I think I mentioned most of these collaborators for this project and some others here, at least here, that we collaborate on some other projects. I think with that, thank you for your attention.
Testing.
I heard you.
Okay, excellent. Great talk. I have two related questions on non-histone acetylation targets of P300. And one is in your either your TAZ2 cancer mutants or in your deletion, in your CRISPR mutant. Do you see increased acetylation of non-histone targets? Presumably, well, not presumably, but be interesting those that are actually in the cytoplasm and not in the nucleus. And the related question is, I'm interested in what's the TAZ domain's regulatory role for non-chromatin-based substrates? Because if you require activation, right, de-repression of this autoinhibitory portion of TAZ2, if it's not a transcription factor in the cytoplasm on non-histone targets, what relieves, where's the de-repression of the TAZ2 domain coming from to allow activation of P300 targets in the cytoplasm, or CBP for that matter? And I guess related is, would TAZ2, so do you think it's transcription factors that are activating non-histone targets? Or do you think TAZ2 has another mechanism of regulation that might involve phosphorylation or binding a non-transcription factor based protein in the cytoplasm, for instance?
Yeah. Great questions. First one, we had a look at a few. We didn't look at cytoplasmic proteins. We look at a few transcription factors, such as PP3.
Right.
We can see increase of PP3 acetylation.
Yeah.
But the increase somehow is not as drastic as nucleosome, using nucleosome as substrate.
Yeah.
So I think the short answer, lead to the answer to the second question is, I think probably it doesn't matter whether it's a nucleosome or not, as long as you have a protein that can somehow bind to TAS2
and disengage it from
deassociate with HAT, then the pocket become accessible to substrate. Right?
Is there any motifs in how transcription factors bind TAS2 that you could then search for that motif in non-transcription factors, for instance, to see if those guys do regulate the activity?
Yeah, there is a motif. I think most or almost all the known TAS2 binding regions, there are transactivation domains of transcription factor. So there are flexible regions, but they contain a few hydrophobic residues. I think the interface is kind of hydrophobic within alpha 1, 2, 3. They're forming a kind of a pocket there, interact. More kind of hydrophobic interaction with. There's a consensus sequence. It's I, X, X, B, X, X. Yeah, the kind of.
It's a hydrophobic, not a acidic, not a.
There are a few acidic residue there, but largely it's hydrophobic surface with 1 or 2 acidic residue somewhere. Yeah.
Okay.
Yeah, it's TADs of TFs.
Okay. I'm going to turn this over to someone else now. Thank you so much.
Thank you.
Yeah, nice talk. Yeah, I really like your study of mechanism, different domain, positive or negatively regulated P300. I wonder at any given time, so how these different domain coordinate to regulate HAT activity, because some are positive regulating domain, some are negative regulating domain. Are these cell context dependent or what? How do they coordinate?
Yeah, I think short answer is you need them all. So most of the HATs, they are in, many other HATs, they are in complex with a few or 10 or 20 subunits. They form complex. Those complex, their function is to regulate the activity recruitment or maybe conformation change of the catalytic domain. But P3 and CBP, they are somehow unique, different from others, is that they themselves are kind of a domain complex. That's why they contain all these domains. They can form this function. Some function as reader of acetylation, some function as reader of unmodified, some function as structure regulatory of the HAT activity. I guess the short answer is you need them all, just like 5 fingers. You need all these 5 fingers to grab a ball, right? That's my understanding. So if you take just one of them, you have compromised activity.
So if you knock down this TAS2, do you affect other domains' ability to modulate HAT? Is what I'm getting. Like you talk about other ring, PhD domain, bromo domain, ZZ domain. So their ability to regulate HAT do you see those change when you knock down the TAS2 domain?
That's a good question. So in the polypeptide we purified without TAS2, it's already a very active enzyme. We haven't, I think there's still some kind of crosstalk between each mechanism, because if you have only the HAT domain, I think it's not as active as the one with Brahma there. I think with different region, only HAT with Brahma, HAT with different domains and the truncation in the different domains, they come up with different activities. I think in that regard, I think each domain they contribute, they somehow crosstalk with each other.
Okay. My other question is related to the importance of the H3 lysine 18 and H3 lysine 27 acetylation in terms of biological process like apoptosis. I understand your reasoning for combining the TAS deletion plus HDAC inhibitor is try to push the limit, maybe enhance further H3 lysine 27 and H3 18 acetylation to a limit when the cell has to go through apoptosis process. But you also mentioned in the ovarian cancer cells, those levels are already very high. In cancer cell, they are already very high. Do you think in that case, they are just high but not reaching the limit? So you have to push it further, use HDAC inhibitor. So if that is, if this is true, you should see those ovarian cancer cells are very sensitive to HDAC inhibitor. Is that the case?
Yeah, that's a great question. That's one of the questions that we haven't addressed in that paper. That's why it got rejected from Nature. Yeah, I think there are a few possibilities. Probably one is that the cancer cell lines, they bearing these truncations, they have some other way to circumvent this high level of acetylation. For example, maybe they have superactive HDAC, or they have some other things, or reduce activity of other has. We don't know yet. There is another possibility is in our case, CRISPR edited cells. 90% of the case is what's that? Not homozygous, two allele truncation. You truncate both allele of this one enzyme. But in cancer cells, they're almost all one allele truncation. They still retain one allele of wild type. So we cannot 100% exclude the possibility that maybe there's some region of downstream of TAS2 that is essential for certain function. So that part we haven't excluded the possibility. So one way to address this, we're now trying to generate the detect system to using cancer cell line targeting each individual allele, either wild type allele or the truncated allele, try to test whether it's the truncated allele or it's the wild type allele required for cell survival. So yeah, that's a great question we haven't addressed. Yeah.
Any additional questions? I have one about, so CBP and P300 can form heterodimers, right? And that's part of, yeah. Do you all see that as part of the mechanism of activation for the HAT activity, that you need to have a homodimer of P300 or a heterodimer of P300 and CBP in order to get efficient HAT activity?
We didn't distinguish whether they are dimer, oligomer, or just monomer, which is, in our case, we just used the purified protein.
Yeah, because based on that literature, it might be interesting to see if you were to make like your TAS2 domain truncated, right? So this normally would be a very hyperactive protein, but you would make a catalytic mutant in that, so that the hyperactive should be now catalytically dead. And then you could ask, does that actually work in trans, with another P300 CBP molecule, or is that a cis mechanism?
I think that's possible because there's one structure showing that in the crystal structure that the two molecules, because the old model is the ALL fold back to inhibit its own activity. But that structure from the same lab, it showed that the two can form dimer with using their own ALL to insert the pocket of another enzyme. Right. So the two can inhibit each other. So I think probably both are possible, because if you think about in cells, P53 probably form this tetramer or even larger complex. And with each one bind to maybe two P300, you can think about the complex, right? So I think either way is possible. Yeah. Right. Yeah.
All right. Any additional questions? Well, let's give Xiaobing a big round of applause.
Thank you all.